When we're talking about cell signaling, there's two main components that we're working with. We have a chemical signal, which is known as a ligand, and then there's going to be some specific receptor that is going to recognize that ligand and help the cell to respond to that ligand. Now, the ones that we're looking at here are specifically the cell surface receptors. So these will be at the cell surface, and we also refer to them as extracellular receptors. The ligands are always going to be binding on the external side. So this is going to be the cell's exterior up here. And just to give us reference, on the bottom is going to be the cell cytoplasm. So as we have it shown here, there are three different types or three different classes of cell surface receptors. So I'm going to talk about the blue one here first. The blue one is going to be what we call a gated channel. And these gated channels are typically for ions, and they are basically a tunnel across the membrane. They allow things to move across the membrane, but it's not going to be unregulated movement. So when we say it's a gated channel, what that means is that these will basically open and close in response to ligand binding. So as I have it drawn here, it's pretty closed up. It would be difficult for some molecule or some ion to actually get through that particular channel. So if I draw some actual ions on here that would want to move through this channel, I've got a high concentration of them on the exterior of the cell. They would love to get to the other side because they do want to move down their concentration gradient, and there's so many more of them on the upper side. But if we have this closed up in the middle, they're going to have a really tough time getting across. So if we did want these to be able to get across, this gated channel can open up if we actually have a ligand bind. So if this is the specific ligand for this particular receptor, upon ligand binding, so here we can see the ligand bound, and once the ligand binds, the channel is going to open. Okay, so I've got my channel bound there, and then if I just erase the middle of it just a little bit here so we can see what would happen. Once it has bound, we would certainly have these little substances being able to actually move through, and they can then flow to the other side until the ligand is removed. If that ligand is removed and we get rid of it on here, then all that means is that we're going to go right back to the closed state. And so if we were to draw the closed state here, it's going to close back up. So we'll see it kind of swoop together in the middle so that we can't get anything through this one anymore. Okay, so that's one type of cell surface receptor. They are the gated channels. They open and they close in response to ligand binding. Sometimes when the ligand is bound, that's when they'll open. Sometimes it's going to be the opposite if we're talking about a different cell surface receptor. But in any case, ligand bound and without ligand is going to be two different versions. One's open, one's closed. We can carefully regulate what's going to be coming in and out of the cell at that moment in time. The second cell surface receptor that we have drawn on here is the purple one, and this is called a G-protein linked receptor. And these G-protein linked receptors are very important when we're talking about signaling with animal cells. They are called G-protein linked receptors because they have a special protein attached to them, which is called the G protein. And that G protein, it does have a couple different subunits that make it up. So I have shown 3 subunits in this one here. So it's the yellow protein that we see stuck on. And typically, that G protein is going to be attached to the internal side or the cytoplasmic side of this particular receptor. And when the ligand binds, so here I have a ligand coming in, as soon as it comes in and actually binds in position, what we're going to see is that binding, so ligand binding, causes the G protein to dissociate. So this means that some of the individual pieces will come off. They will actually separate from the others. So we're going to see this separation here. And when we get that separation, there will be parts of the cell that are able to see that. They can interpret that dissociation. And that is actually going to trigger some type of a response on the inside of the cell. So once that dissociates, we get a response. And that response that we have is ultimately going to be a signaling cascade. And as a signaling cascade, we'll have a lot of different things happen from that point forward, where you basically have one protein or substance that's going to recognize that something changed, and it will tell or signal that to another neighboring molecule. And we'll probably have involvement of a lot of secondary messages or secondary messengers that occur down the line. So again, the G-protein-linked receptors, you have a G protein stuck onto the receptor on the cytoplasmic side. It is going to come off when the ligand binds, and that coming off or that separation is going to cause a cellular response. The third type of receptor that we have on the cell surface is what we call an enzyme-linked receptor. And enzyme-linked receptors, we have a lot of different really categories of them or types of them. But the main idea when we're talking about the enzyme-linked receptors is that the internal portion of that receptor has enzyme activity. And that enzyme activity will be activated when you have a ligand bind to the outside surface of the receptor. So if we draw that happening here, so if we had a ligand bind up here, that binding on that surface is going to cause some kind of overall shape change in our molecule. Okay, so it will be altered in some way so that basically that activates the enzyme portion on the other side. And when that enzyme portion is activated, it is going to be able to do whatever that particular enzyme is designed to do. Now, in many cases, that is going to have kinase activity. And when we're talking about kinase activity, kinases have the capability to add phosphate groups to things. So there may be some phosphates down here, and as soon as we activate this portion, it's going to be able to take those phosphates and maybe attach them to some neighboring molecule right here, so that this molecule, which previously was not phosphorylated, now it actually has the phosphate on there. So many of these do act as kinases. It's not always going to be the case, but kinase activity associated with those enzyme-linked receptors is going to be very common. So ligand binding is going to cause oftentimes some type of phosphorylation, or this could cause dephosphorylation as well. And then after that happens, that molecule here is mobile, and that can cause a signaling cascade inside the cell. And once we get that signaling cascade, hopefully we get that ultimate response that we are looking for and that the whole ligand was trying to cause inside the cell.